Welcome back to Biomandscape. Here, what is a hormone? Hormone is a signaling molecule that is produced by one cell or a tissue that can cause a response in another cell. It acts as a messenger. Then, example like auxin, gibberellin, abscisic acid, ethylene, cytokinin, brassinosteroid, and peptide hormone like CLV3, cysteamine, and PSK. So here, the hormone it binds to a particular receptor. It is like a key that carry the information, and receptor is like a lock where it is binding. So receptor is a protein that specific recognize and bind the hormone, and once the hormones bind the receptor, initiate a signaling cascade inside the cell.
Auxin binds to the TIR1 receptor. This promotes the degradation of AUX proteins, directing gene expression and plant growth.
Here we can see the visualization where the TIR1 it binds to the auxin receptor, GID1 binds to the gibberellin receptor, PYR and PYL it binds to the ABA receptor, that is abscisic acid, and BR1 it binds to the brassinosteroid receptor, and CLV1 it binds to the CLV3 peptide receptor, and pheronia it binds to the RALF receptor. So what is a repressor? A repressor is a protein that blocks the gene expression or prevent a signaling pathway from being activated. So it is often removed or degraded when an hormone is present. It acts as a stop on the gene expression. Like DELLA protein repress the Gibberellin signaling, Auxin and IAA protein repress the auxin responsive gene, and Jas protein it repress the Jasmonic acid signaling. So the repressor it is like a brake of a car. When the hormone signal arrive, the brake is released so the response can occur. So first let's know the auxin signaling. Auxin signaling is a beautiful example of a single dependent gene regulation. It is unique. It have a unique feature of the pathway in auxin does not directly activate the gene. Instead, it removes a repressor, allowing the transcription to occur. So if there is no auxin present, then what will happen? Inside the nucleus, the ARF or auxin response factor, that is a transcription factor that is capable of activating the auxin responsive gene, the AUX and IAA protein, it will bind to the ARF and prevent it from functioning. So what will be the result? The ARF cannot bind the DNA effectively, so auxin responsive gene remains off. So ARF is like a driver, and AUX and IA that is a handbrake. So the driver is ready but the brake is on. If auxin enters the cell, what will happen? If auxin diffuse into the cell and reaches the nucleus, here there will be binding of TIR1. And what is TIR1? It is a F-box protein and a part of the SCF, ubiquitin ligase complex. That is, its job is to identify the protein that need to be destroyed. Then auxin, it will act as a molecular glue. It is most important thing. Here auxin bind to the TIR1 and AUX and IA increases their affinity for each other. So auxin act as a molecular glue, allowing the TIR1 to recognize the AUX and IA. Then the next step is ubiquitinylation of the AUX and IA. Once the AUX and IA bind to the SCF TIR complex, the ubiquitin molecule that are attached to the Auxin IA, and this process is known as the ubiquitination. So this think about it as destroy me target. Then proteasomal degradation happens. The ubiquitinated AUX and IA protein is recognized by the 26S proteasome. The proteasome degrades AUX and IA into simple peptide. Now the repressor is gone. Then ARF become active, and without the AUX and IA blocking it, the ARF bind to the auxin response element, that is AUXRE, in the promoter of the target gene. So the RNA polymerase is recruited and transcription begin. So AUX responsive gene are now expressed, and these genes regulate the cell elongation, root initiation, vascular differentiation, tropic response, and embryogenesis. So why is this path efficient? Instead of directly activating the gene, auxin eliminate and inhibit the AUX and IA, and it allows the plant to respond rapidly to change in the auxin concentration. Now next is the gibberellin signaling, where the gibberellin is produced hormone, then it will bind to the, 'GID1 receptor, and the GA, 'GID1 complex is bind, then it will activate the gibberellin receptor, and it will bind to the auxin and IA. The repressor, it binds the DELLA, leading to the DELLA degradation, and that result in the growth promoting gene as that are expressed. So GA will bind to the GID1 receptor, then DELLA degradation will occur, and the growth gene is on here. So here we are going to know about the Jasmonic acid signaling. So Jasmonic acid is a lipid derived plant hormone. The biological active is JA isoleucine, that is jasmonyl isoleucine, and it regulates the defense against insect and pathogen, wound response, fertility, and stress adaptations. So here we can see the structure of jasmonic acid. Then the JA signaling works by removing a repressor JAS when the jasmonic isoleucine is absent. The JAS protein blocks the activator MYC2, and when the jasmonic acid isoleucine is present, the JAS that is degraded to MYC2. MYC2 is free. So and defense gene they are turned on. So here we can see when is the gene off and when is the gene on. If there is no jasmonic acid, the JAS will bind to the M.Sc2 MYC2 and the gene will be off. Plus if the jasmonic isoleucine is present, jasmonyl isoleucine is present, so what will happen? The gene, the MYC2 will be binding to and the gene will be on. So the key component is the jasmonyl isoleucine is an active hormone. The CoI1 is a receptor in the F box. The JAS is a repressor protein. The MYC2 is transcription factor. The SCF-CoI is E3 ubiquitin ligase complex, and ubiquitin is present and 26 proteasome that degrades the JAS. So here we can see a diagram that shows if there is no jasmonic acid present, the gene will be remaining off. So JAS is an inhibitor of MYC2 and this will off the gene. So in the absence of jasmonic acid, JAS protein bind to the MYC2 transcription factor. The MYC2 cannot activate the target gene. Its defense genes are off. If wound or stress occur, the insect attack the pathogen infection, or mechanical wounding trigger the jasmonic acid biosynthesis in plants. And if there is a jasmonyl isoleucine that is synthesizing here, so the active hormone that is jasmonyl isoleucine is rapidly produced and moves into the nucleus, and then the jasmonyl isoleucine bind the CoI1. The jasmonic isoleucine bind to the receptor of CoI1 and F-box protein that is a part of the SCF-CoI complex. Then what will happen? It will jasmonyl isoleucine act as a molecular glue that promotes the CoI1 to bind the JAS protein. Then JAS is ubiquitin-mediated and degraded. So here what is happening? The SCF-CoI1 ubiquitinate the JAS protein, and the ubiquitinate JAS is degraded by the 26S proteasome. Then the MYC2 is free and activates the gene on. So here we can see with the JAS gone, the MYC2 is released and it binds to the promoter of JA responsive gene and activate the transcription. So what is the result? Activation of the gene can lead to the production of proteinase inhibitor, secondary metabolite, antimicrobial component, volatile organic component, and other defending proteins. So outcome is enhanced resistance to insect pathogen and better stress tolerance. So first is wound healing, then jasmonyl isoleucine synthesis occurring, then jasmonyl isoleucine plus CoI1, then it will bind to the SCF-CoI binding the JAS, and JAS ubiquitinylation and degradation occur, and then MYC2 will be released, and then defense of the gene will be on. So important for examination point is the active form of jasmonic acid is jasmonyl isoleucine and not a free jasmonic acid. The CoI1 is a F-box protein receptor. The JAS protein are repressor of jasmonic acid signaling, and the SCF-CoI is an E3 ubiquitin ligase complex. The JAS degradation release the MYC2. MYC2 activate the JAS responsive gene, and the jasmonic acid signaling is important for the plant immunity and stress response. Let's know about the plant hormone and their repressor. Auxin is having a major repressor, Auxin IAA. Gibberellin is having a repressor, DELLA protein. Jasmonic acid is having a repressor, JAS protein. Spironolactone is having a repressor, SMXL, that is D53 protein. Brasinosteroid is having a repressor, BIN2 kinase. ABA, abscisic acid, is having a repressor, PP2C phosphatase. So these are the major repressor of plant hormone. Thanks for watching. Do like, share and subscribe Biomindscape.